This video will focus on chromatin remodeling as a form of gene regulation in eukaryotes. In a previous video, we discussed the role of histones in creating areas of the DNA that were accessible for transcription, the euchromatin, and areas not generally accessible for transcription, heterochromatin. The structure of chromatin as euchromatin or heterochromatin is dynamic, and these changes have important implications in gene expression. Changes in chromatin structure can range in size and involve changing the configuration of chromatin to either closed, which is tightly packed, or open configuration, which is accessible for transcription. Changes in nucleosome position facilitate differential expression of genes. Many transcription factors that activate this process act on nucleosomes to change chromatin structure. An example is the beta globulin gene, where changes in the nucleosome position in the promoter region activate transcription. One way that this happens is through ATP-dependent chromatin remodeling, which, as the name suggests, uses energy to change the location and/or the composition of nucleosomes, thereby affecting the rate of transcription. A main player in this process is DNA translocase that helps change the conformation of the chromatin. But there are also many additional families of chromatin remodelers that we will sadly need to leave for a different discussion. In our cells, there are three main ways chromatin remodeling complexes change chromatin structure. So let's take a look at them. The first is change in nucleosome position. In the most basic sense, this action changes the relative positions of nucleosomes to each other. This can be through a variety of actions, which we will touch on in a moment, but suffice to say, this process can cause the nucleosomes to become more spaced out and therefore more open to transcription. The second way is through histone evictions. In this process, whole histone octamers are evicted from the DNA strand, leaving stretches freely accessible for transcription. The third and final way is through changing the composition of histones by inserting histone variants. This one is a bit more complicated and warrants just a smidge more discussion here. Histone variants are mutated histones that behave a bit differently than standard histones. Standard histones have a role as either a core or linker histone that serve to bind DNA or interact with other histones. Histone variants change that action slightly. There are several variants, so we won't dive into them all, but one example, histone H1's variant shown at the top of this table here, that forms a tighter association between histones and is associated with gene repression. These histone variants tucked into the octamers can change the conformation of chromatin and create specialized types of it. Histones can also be modified in a variety of ways, especially on their amino terminal tails. Common modifications include acetylation, methylation, and phosphorylation. All of these affect how nucleosomes interact with each other or with the DNA. For example, acetylation, shown here, leads to less tightly bound DNA to the histone octamer. These different modifications serve as markers and binding sites for various proteins that alter chromatin structure. This language of histone modifications that these proteins recognize is called the histone code, and this has large effects on transcription.